Hello everyone, welcome back to my channel Pari's tutorial. I'm back with one more interesting video. So let's get into the class. Good morning everyone. Welcome back to my channel Pari's tutorial. Today I want to talk about the replication of DNA, how the DNA copies have been produced by semi-conservative mode of replication. Okay, so before going to start the replication, last class you have already studied about the structure of DNA, the arrangement of nucleotides, the arrangement of sugar phosphate backbone of DNA, and how according to Chargaff's rule the whole nucleotides have been arranged in DNA molecule. DNA replication was started by Watson and Crick. There are many modes of replication, out of which only the Michelson and Stahl have proved that DNA replicates through semi-conservative mode of replication. So, what is replication? So, replicating is producing more number of copies of itself during the life cycle of an organism, right? The genetic material has to be replicated, has to be produced, has to produce large number of copies in in a single organism for it to continue its genetic material to be transferred to the next generation, right? So DNA replication is a must process that happens in every living organism. Today, what are the possibilities of possible modes of replication? Let's discuss. See, there are three modes, out of which this is considered to be the correct one. Okay, in dispersive mode, the parent DNA molecule is made into made many cuts, out of which they possibly form a daughter DNA, but it is not acceptable. Okay, and for the conservative mode, the whole DNA parent molecule would be moved into one daughter molecule, and the other daughter will be having the new strand. One daughter will be having the same old strands of the parent DNA. So even this is not accepted. Finally, the semi-conservative mode was being proved by an experiment where they have used a density gradient centrifugation. The next topic I will talk about what is density gradient centrifugation and how the process has occurred. Okay, so let's talk about what is semi-conservative mode of replication. The semi-conservative mode of replication, in which one strand of the parent DNA would enter into one daughter molecule. In the same way, this other strand will get the other strand. Okay, so and this particular strand of parent molecule will act as a template. So it produces one old strand and one new strand in daughter molecules. So this was actually done on E. coli. So you should have heard about Escherichia coli, right? These E. coli organisms are made to grow on a medium which contains N15. N15 is a isotope of N14. Okay, it's a heavier isotope, right? So they are grown on that. So all strands of DNA of that particular E. coli are now labeled with N15 isotope. Okay, so see, this is the N15 heavy isotope that I am talking about. So they are made to grow on the isotope for one generation. Okay, so what they have done is strands of DNA now are labeled with N15 isotope. Now these DNAs are being isolated and they are made for centrifugation, because N15 isotope being a heavier isotope than N14, it would form a band with a heavier band, right, at the lower part of the tube, right? So in the upper part, the N14 is the lighter isotope than N15, so it forms a lighter band. So they get two bands, right? So what happens after the all the DNA of E. coli are labeled with N15. Now they transferred that medium. They transferred that particular E. coli labeled with N15 to a medium containing N14. So they are made to replicate for 3 generations, like should have for 3 cycles. So first it is now N15 term, complete N15. Once they are grown on N14, one strand of DNA of parent would enter into each daughter cell, the other strand would enter into the other daughter cell. As they are growing in the N14 medium, they will accumulate that particular N14 to it. So they contain both N14 and N15 in the first generation. And after that, the second generation, again this N14 N15 would again divide into N15 and N14, and again it takes the N14 isotope that was bare grown, so and again it will get N14 N14. So as you are keeping this particular DNA in N14 medium, the DNA gets more and more lighter, because they are first at N15, they are at a heavier band. So they become lighter and lighter as they are gone on N14 medium. So after the three modes of generation, of three modes of replication, most of the DNA are between the lighter band and the heavier band. So first they are grown in the heavier band, then they are slowly moving towards the lighter band, because that particular DNA is being separated to the daughter cells equally. So this N15 gets separated to N14 and N15, again N14 and N15, finally N14 and N14. So everything relays between the two bands, and finally even if you keep those cells even more for N14 medium, they keep going towards your lighter band. They started from the dark band, and they have moved towards the light band, which says that the N15 isotope is being distributed to equally for both of them, both the cells. Then they are going to the as they are kept growing only in the N14 medium, the progeny obtained that particular N14 only. So the N15 is being slowly distributed to the progeny. So this says that the DNA that is present in the parent, that is N15, is equally distributed to both the DNA of the daughter cells. So this is the proved method, and for this what they have done is the whole process is kept in a centrifuge for a speed of 30,000 to 50,000 rotations per minute. Okay, I will tell you what are rotations per minute in centrifugation. Okay, so 42 to 72 hours. You all know that what is density gradient centrifugation, so I will also discuss that in the next class. So based on the density of cesium chloride has got the same density of DNA and RNA, so that is being used for the centrifugation of this particular DNA. So at 30,000 to 50,000 rpm for 42 to 72 hours, they got two different bands, one light band at the top and N14 top at the top and N15 at the bottom, as it is a heavier isotope than N14. So N15 is a darker band which is present at the end. This is the N14 which is present at the top. So the middle bands are the lighter bands that have been becoming lighter and lighter because of N14 getting distributed to all the daughter progeny. Okay, see, the parent molecules have got their DNA labeled only with N15. Okay, so when you go for the band, you will get a darker band which is a heavier band. Okay, in the same way, in the first mode of replication, what happened? They are grown in N14 medium. Because of that, one progeny got one daughter got N14 and other daughter got N14 and N15. It's a hybrid, like, right? So they got one from N15 parent and other from the medium they were being replicated, right? So it's an intermediate thing. So this is neither it is neither dark nor light. It is in between the N15 band and N14 band. So on the second side of replication, what happened? The N15 is even more distributed. So because of this, this N14 has become lighter and lighter and lighter. So it is being moving towards your lighter band, right? So after the third mode of replication, you can see how the progeny were being distributed. Out of four, only one progeny has finally got this N15 because of its replication. So this says that based on the centrifugation, based on the bands that they got through centrifugation, they got to know that the DNA is being distributed equally from parents to both the daughter cells. Okay, this is what you call a semi-conservative mode of replication. Okay, so this topic is about replication. Okay, and I want you to get to know that what are the enzymes involved in the process of replication, which is very important, because they may ask you what are the enzymes required for formation of replication fork, what are the enzymes that help you to bind and unwind the DNA during replication. Okay, so let's start the replication process, and I'll tell let's start how the replication starts. So first of all, DNA has to, it's a 5 prime and 3 prime end, right? So it has to be first of all separated. For one strand to act as a template, the other strand to act as a template, the first DNA has to be separated. For that, there are two important enzymes called gyrase and helicase would bind to the DNA. Once they bind to the DNA molecules, the two templates of DNA would get separated. So this is what you call DNA unwinding or DNA melting. The whole thing will be DNA melting. So I will tell you, see what happens. This is my diagram that I thought I could explain to you in a much better way. See, this is your 3 prime end of DNA and this is your 5 prime end of DNA. Okay, so each and every DNA has got origin of replication, ORIC site. Okay, where the initiation has starts. Now this is a DNA helicase that would help in unwinding. So once they have unwinded, say this is called the replication fork. It resembles fork, so it is called a replication fork. Once the DNA has been separated or melted, immediately SSBP, they are called single standard binding proteins. Okay, these particular single standard binding proteins would immediately come and attach to 3 prime side and also the 5 prime side of your DNA fork, because these strands have to be stabilized in single standard only. So they are called single standard binding proteins. They would help you to maintain these two strands in a single standard only, because they have to act as a template for producing the second strand. Okay, so this is what you call single standard binding proteins. So once they are ready for that, the immediate thing that comes is primase is an enzyme, okay, that produces RNA primer. See here what happens is primase produces an enzyme called primase produces RNA, primer RNA. This primer RNA starts with 5 prime to 3 prime direction. So this is being in 3 prime direction and this is being in a 5 prime direction. So the replication starts with 5 prime to 3 prime direction towards your origin. Okay, it is going towards your origin and this is helped by a primase enzyme. This particular primer RNA has got a free 3 prime OH group, right? This particular black thing is a 3 prime OH group. This 3 prime OH group is an activity site for initiation of your DNA polymerase enzyme. Okay, this particular DNA polymerase enzyme would enter for it, DNA polymerase enzyme needs a free 3 prime OH, so which is present on the primer RNA. So once the DNA polymerase attaches to this primer, it starts producing your DNA further. Okay, and this is continuous process which occurs from 5 prime to 3 prime direction towards your origin. So this is your leading strand. Okay, the 3 prime to 5 prime direction where the template, this acts as a template, and where this strands keep on producing, and this is called a leading strand. Okay, so in the same way, once this has started, this keeps going here and this is called leading strand. DNA polymerase would continuously help you to add all polydeoxyribonucleotides to your 3 prime side. So this is your 5 prime side, this part is your 5 prime side and this part your 3 prime side. The DNA polymerase would add all polydeoxyribonucleotides continuously to form your daughter DNA. And now after this is done, the primer now starts with in this direction. the same way whatever the primase that has started here, the same primase would start here and move towards here for the formation of the lagging strand replication. So this is your lagging strand. Okay, this is your lagging strand, and it would continuously give rise to small fragments of DNA, because here it is a discontinuous mode of replication. Okay, see, this primase, every time the primase has to come, add the primer DNA, and DNA polymerase should start, and polymerase keeps adding polydeoxyribonucleotides, and again it will get digested, and again the primase will come and add the primer RNA, and primer RNA would attract the DNA polymerase to attach to it, and DNA polymerase again add all the polydeoxyribonucleotides, and again it get digested. In the same way, this is how the primase should always come to this a lagging strand replication and keeps on and on. Okay, that is why this is not a continuous strand. It has to come, arrange, and should disappear. So that is why you will get small fragments of DNA, and this is called Okazaki fragments. Okay, so these are all called Okazaki fragments. At the same time, there is an enzyme called ligase. Okay, there is ligase here. The ligase would ligate. Ligation is nothing but attachment, right? So the DNA polymerase, once the whole thing is done, the primer is being digested by the DNA polymerase itself. Okay, RNA primer is being digested by the DNA polymerase, and you got your complete second strand, right? In the same way, the digestion primer, and all the time whenever the primase comes and gives the primer, after that the digestion is also taking place in the lagging strand replication. So you will get all small Okazaki fragments, and all the Okazaki fragments of the lagging strand gets ligated by DNA ligase. So that is how all the enzymes would play important role in the formation of a replication. So helicase, gyrase play unwinding part. Single stranded binding proteins would help in stabilizing those single stranded DNA. Primase would help to produce primer RNA, and hence attracts the DNA polymerase for further polymerization. Then that produces leading and lagging. After that, digestion of primer takes place by the DNA polymerase. Once they are once they are once they produce the DNA, the digestion primers would be digested away, and later all the Okazaki fragments that have been produced were being ligated by DNA ligase. So finally you got your progeny separated, right? This is what the replication is all about. Okay, so what are the proteins that are involved in DNA replication? So I already told you importance of all these things, and apart from all these things, the main role are DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. So why is DNA polymerase important? DNA polymerase not only does that polymerization activity in 5' to 3' direction, it has also got an exonuclease activity, which we can discuss in the repair mechanism. So what happens is, when to you about the DNA repair mechanism in detail in the next class. Okay, so as of now, I thought till now you have got to know about the type or the structure of DNA, its arrangement, its nucleotide arrangement and all, and what is the replication. Class we have covered the main important biomolecules in detail. Okay, let me know the feedback about it. Thank you so much for listening to my class. So please like, share, and subscribe to my channel.